Oh, yeah. It's a beautiful morning, and the science gods have blessed us with a freaking cool paper. So, I don't know when this came out. Oh, wow. It got posted, okay, yesterday. Using a GPT-5 driven autonomous lab to optimize the cost and titer of cell free protein synthesis. Okay, so my understanding of what happened is GPT got access to some library that allowed them to write programs on a Ginkgo autonomous rack lab system. That's my understanding, cursory. But let's just read it. Because I just want to read the whole thing, because it's only going to be like 20 pages. We used an autonomous lab comprising a large language model and a fully automated cloud laboratory to optimize the cost efficiency of cell free protein synthesis. By conducting iterative optimization, the LLM-driven autonomous lab was able to achieve a 40% reduction in the specific cost dollars per gram of protein, which I love that. That's one of those unit of measurements where you're just like that's like flop per dollar, where you're just like or dollars per flop. Anyways, it's like a great unit of CFPS, so cell-free protein synthesis relative to the state of the art. This cost reduction was accompanied by a 27% increase in the protein production titer. So what does that mean? Grams per liter? I didn't know that. I mean, I guess I should have looked at grams per liter. Okay, it means concentration. Iterative experimental design, experiment execution, data capture and analysis, data interpretation, and new hypothesis generation were all handled by the LLM-driven autonomous lab. The interface between GPT-5 and the lab incorporated built-in validation checks via pedantic schema to ensure that AI-designed experiments were properly specified. Yeah, see, this is where I would love to use lean. I mean, you could use pedantic, but why not use lean, bro? Experimental designs were translated into programmatic specification of multi-instrument biological workflows by Ginkgo's Catalyst software and executed on racks. So this is kind of like where I'm hoping to get some more insight. Is they were they were they automatically were the programs generated automatically, or did someone have to go in there and click a bunch of buttons? With human intervention largely limited due to reagent and consumables preparation, loading and unloading. Wait, hold on. With human intervention largely limited to reagent and consumables preparation, loading and unloading. See, this is where when I was talking about what I would spend $50 billion on, you would not have this. By integrating LLMs with programmatic control of a cloud lab, we demonstrate that an LLM-driven autonomous lab can successfully perform a real-world scientific task, highlighting the potential of AI-driven autonomous labs for scientific advancement. There are two central bottlenecks in scientific research. The first is so-called wet lab experimentation, the hands-on physical manipulation of chemical and biological materials to conduct experiments, and the second is intelligence, the conception, planning, and design of experiments, and the analysis and interpretation of resulting data. To answer a given scientific question, advances in lab automation, software architectures, application APIs, and cloud labs promise to alleviate the former bottleneck. Similarly, rapid advances in LLMs, particularly reasoning models that can solve complex multi-step problems by breaking them down into logical sequential steps, can automate the latter. Together, reasoning models and autonomous labs are capable of performing all steps of routine scientific discovery: experimental planning, design and execution, and data analysis and interpretation, and new hypothesis generation. Early efforts have illustrated the potential of autonomous labs across multiple disciplines, including chemical research, material science, and biological design. Until recently, reasoning LLMs, have been deployed mostly in computationally verifiable tasks such as algorithms, math, and physics. Evaluating AI performance in designing biological experiments is particularly challenging due to the need for wet lab validation. As a result, yeah, it's like, what are you going to do? Have an AI design an experiment and then the humans are the freaking slaves? What? That doesn't make any sense. As a result, the potential of AI-driven autonomous labs for advancing real it remains largely untapped. Totally agree. One such real-world scientific application is cell-free protein synthesis, a method for recombinant protein production of both laboratory and commercial importance that has proven difficult to optimize. Oh man, we got to go through these sources. Given the widespread use of CFPS in rapid prototyping, including antibodies, peptides, proteins, enzymes, and proteins for structure determination, as well as commercial production of an antibody-drug conjugate, luveltamab, tazevibulin, holy, luvelta. Improvements in self. What if we just go to the Wikipedia page and get some actual? Okay. Cell-free protein synthesis, CFPS, is the production of protein using biological machinery in a cell-free system, that is, without the use of living cells. The in vitro protein synthesis environment is not constrained by a cell wall or homeostasis conditions necessary to maintain cell viability. Thus, CFPS enables direct access and control of the translation environment, which is advantageous for a number of applications, including co-translational solubilization of membrane proteins, optimization of protein production, incorporation of non-natural amino acids, selective and specific site labeling. Due to the open nature of the system, different expression conditions such as pH, redox potentials, temperatures, and chaperones can be screened. Since there's no need to maintain cell viability, toxic proteins can be produced. Oh, that's actually cool. I didn't think about that. Common components of a cell-free reaction include a cell extract, an energy source, a supply of amino acids, cofactors, DNA with the genes. A cell extract is obtained by lysing the cell of interest and centrifuging out the cell walls, genome, and other debris. The remains are the necessary cell machinery, including ribosomes, aminoacyl tRNA synthetases. Yeah, we just learned about these. Right. These are the things that attach the amino acid to the tRNA. Okay. Translation initiation of an elongation factor, nucleases, etc. Two types of DNA can be used: plasmids and linear expression templates. Plasmids are circular and only made inside cells. LETs can be made much more effectively via PCR, which replicates DNA much faster than raising cells in an incubator. While LETs are easier and faster to make, plasmid yields are usually much higher in CFPS. Because of this, an energy source is an important part of a cell-free reaction. Usually a separate mixture containing the needed energy source along with supply of amino acids is added to the extract of the reaction. Phosphenolpyruvate? Acetyl phosphate? Why don't they just use ADP and ATP? CFPS has many advantages over the traditional in vivo synthesis of proteins. Most notably, a cell-free reaction, including extract preparation, takes 1 to 2 days, whereas in vivo protein expression may take 1 to 2 weeks. CFPS is an open reaction. The lack of cell walls allows direct manipulation of the chemical environment. Samples are easily taken, concentrations optimized, and the reaction can be monitored. In contrast, once DNA is inserted into live cells, the reaction cannot be accessed until it is over and the cells are lysed. Another advantage to CFPS is the lack of concern for toxicity. Some desired proteins and labeled proteins are toxic to cells when synthesized. Since live cells are not being used, the toxicity of a major application of CFPS is incorporation of unnatural amino acids. The openness of the reaction is ideal for inserting the modified tRNAs and unnatural amino acids required for such a reaction. Why would you need that? I don't really get that one. Oh, what a weird sentence to see in Wikipedia. Synthetic biology has many other uses and is a bright future. Who the fuck wrote that? Protein evolution, nanomachines, nucleic acid circuits, and synthesis of virus-like particles for vaccines and drug therapy. Okay, let's go back to the paper. The very large CFPS parameter space suggests that its optimization would be a suitable and challenging testbed for autonomous lab experimentation. CFPS reactions consist of many components: DNA template encoding the protein of interest, cell lysate, nucleic acids, amino acids, energy sources, cofactors, coenzymes, buffers, salts, etc. CFPS also produced proteins bearing a wide variety of modifications, including disulfide bonds, glycosylation, I don't know what that means, glycosylation, phosphorylation, and non-canonical amino acids. Further broadening the... Actually, never mind. I'm totally wrong. Using non-canonical amino acids sounds amazing. I'm actually so into that. That's sick. That's my favorite one. Accordingly, there have been a significant prior efforts leveraging machine learning and similar techniques to optimize CFPS reactions against two common figures of merit: titer and specific cost. Reducing specific cost, a critical factor given the much larger scale of experimentation made possible by autonomous labs, without sacrificing titer, makes the CFPS optimization a non-trivial problem. Okay, so GPT-5 with RACs perform autonomous lab experimentation with the objective of minimizing E. coli CFPS specific cost. The LLM-driven autonomous lab was able to produce a standard benchmark protein, superfolder GFP, at a cost of $422 per gram versus a recent state-of-the-art report of $700 by Olson et al. In 2025. This cost reduction was accompanied by an increase of 27% in titer versus state-of-the-art, confirming that cost and titer can be improved simultaneously without sacrificing one for the other. Results. Our approach to GPT-5 driven autonomous lab experimentation applied here to CFPS optimizer is illustrated in Figure 1. We used GPT-5 to design E. coli lysate-based CFPS reaction compositions that optimize production of a test protein, SFGFP, with the objective of minimizing specific cost. The first iteration of CFPS reactions were designed by GPT-5 without the benefit of any example reactions or prior experimental results. A setting referred to as zero shot. Thus, GPT-5 only leveraged intrinsic knowledge stored in model weights to generate experimental reaction compositions. GPT-5 designed experiments that were constrained to be properly specified with this schema thing. And physically executable. That's another thing. All CFPS reactions were run on rack. I mean, how many times are they going to write this? Racks are a modular laboratory automation solution developed by Ginkgo. It's like an advertisement. Each rack consists of a single instrument type, a liquid handler, centrifuge, incubator, plate reader, and standardized hardware that enables a rack to move samples in an SBS format. I don't know what that means. To and from that instrument via 6-axis robotic arm, and between racks via a sample transport track. Racks flexibly combine into unified assemblies to perform fully automated scalable workflows. Bro, this literally is like straight out of there. Ginkgo's rack command and control software Catalyst enables programmatic specification of multi-instrument workflows and orchestrates the running of many such workflows simultaneously on the same rack assembly, including rack and instrument unit operation, sample transfer, and metadata. Okay, I don't even know what that means. Includes all the instrumentation. Supplemental figure 2. I mean, these are the diagrams that I hate seeing. I mean, this is not so bad, but it's like boom, boom, boom. Reasoning, hypothesis generation. Okay, it's just because they're vague. Oh, the fluorescence measurement, that's cool. I want to see figure 2. Supplemental figure 2. Is it not in here? Wait, I feel like I just saw. There we go. I knew it. My boy Will. I emailed him. He didn't respond. That's okay. I'm not bummed out by that at all. This is so cool, man. You can buy adenine for $10 a liter? That's crazy. Oh, in this molar. Yeah, okay. Pretty low concentration. Oh, here we go. Here's a picture of Catalyst. Dude, I've wanted to use Catalyst for so long, and look at that program, man. That is so cool. Example schedule of multiple concurrent Catalyst protocols runs. Come on, guys. What? CFPS reaction optimization workflow. Rows correspond to Ginkgo's Cloud Lab instruments. Within their respective racks, okay, so each one of these rows is an instrument. What does that say? Each color corresponds to a Catalyst protocol run for a single 384 plate. Each colored rectangle denotes occupancy of a given rack. Yeah, that's what you would expect. Some racks, like the large capacity static incubator racks, as well as the sample transport racks, can support multiple simultaneous. Oh, okay. So this is probably like we have 10 spots for incubating plates in this incubator or something like that. I don't know. Oh, here we go. Here's the rack, dude. That's so cool. I wonder how they rendered this or did they photograph it? That is so cool. I love that it's like a really low resolution photo too, so you can't really read any of it. Okay, let's keep going. Where are we? Oh, here we are. OpenAI proposes a batch of experimental designs and validates them. Designs are then programmatically converted to Catalyst protocol runs. Okay, so they do have some sort of compilation thing. After experiments are executed, okay, yeah, then it closes the loop. That's cool. We ran CFPS reactions at 20 microliter scale and 384 plates. To improve dispensing accuracy and precision, different Cloud Lab liquid. I think all they're saying here is like, I don't really know what they're saying there. What does that mean? Oh, okay, yeah. They matched each reagent to the dispensing tool that can handle it best. Some instruments are great at sub microliter. Yeah, okay, I mean, that's important. I would expect you to have to write that. Each plate consisted of 78 distinct reaction compositions. Okay, oh, okay, so 4 replicates each. Purified SFGFP for absolute protein quantification and positive and negative controls. Based on dispense volume and component viscosity. That's actually really, really cool. That's one of those problems that I would love to work on. Each plate consisted of 78. Oh, okay. So that's not, yeah. Oh, okay, okay, wait. So, yeah, so they would Okay, yeah. So that's an interesting uh okay. The Cloud Lab performed reaction component addition, CFPS reaction incubation, and fluorescence data acquisition. We used custom software code. We used custom software code. Yeah, bro. All my friends love software code to translate designed experiments into Catalyst protocol runs. Yeah, actually, that's what I want to look at. I love software code. Figure 2, figure 3. SDS page. Did they automatically do the SGF? We've been doing tons of SDS pages in my class. Okay, I don't really see the because what am I looking for? I'm looking for. All right, so this should explain. Schematic overview of the CFPS reaction optimization workflows as implemented in Catalyst. We implemented the CFPS work as 3 separate Catalyst programs for flexibility. Okay, retrieve reagent source plates. Thaw acoustic dispense reagents into base buffer pre-filled destination plates. Dude, the echoes are so cool. Unfortunately, I went to this conference and they didn't have an echo there. They brought like their liquid handler. It's like, dude, the echoes. I want to see freaking sound. I want to know like how hard really would it be to build a tiny little water gun like that? Like, how do you dude? I freaking love the echo. It's so sick. Bulk dispense 11 microliters of PBS backfill and SGP standard destination. Okay, so these are okay. Okay, so these columns 1 through 3, these are our standard. These are the test. Oh, these are the positive controls and the negative controls. So presumably the positive control. I don't know what would the positive control. I'm glad they said that. Okay, positive control. A known good CFPS mix. Okay, yeah. A known good, okay, and then no DNA template. All right, nice. That makes sense. So that's why there's so few columns in the negative control, probably. But the positive control makes sense, too. Okay, this didn't explain what I was hoping it to explain. I wanted to see their custom software code. Experimental data was returned to GPT-5 via a JSON file comprising protein titers. Human intervention in lab experiments was primarily limited to preparation, loading, and unloading of reagents and consumables on and off the rack system. I would also like to see how that's done. Like how, because obviously any system like this is going to have inputs and outputs that, like, you know, at some level humans need to look at. So I'm just curious how that goes down. The compelling capability of LLM-driven autonomous lab is its ability to generate human-readable laboratory notebook entries. GPT-5 documented how it analyzed data, observations it made of experimental trends, surprising or confusing results, and new hypotheses it formulated for testing. That's actually so cool, dude. Early entries from step 0, for example, critiqued titer variability between replicates within a plate. To reduce titer, what is CVs? Oh, coefficient of variation. Okay. To reduce titer CVs, we modified certain reagent stocks to improve the precision and accuracy of liquid dispensing steps. For example, we reduced concentrations to enable more accurate liquid transfer on the echo and changed stock solutions to reduce precipitation. Switch to a maltodextrin with a shorter chain length. Increase the pH of the tyrosine stock solution to pH 12. Ginkgo personnel performed quality testing of precipitation and echo liquid handling of different reagent stock solutions manually. With these improvements, we were able to achieve median per plate CVs of 17%, with 90% of initially screened plates having CVs of less than 30. So it was greater than 40, and then they got it down to 30. It's still pretty high. In step 0, 146 plates totaling 7,500 unique reaction compositions were designed, and that's just step 0. That's so freaking cool. In step 1, GPT-5 used experimental data from step 0 to inform the next round of reaction designs. 32 plates were executed on the lab with data returned. In this step, GPT-5 was able to design reaction compositions with reduced cost but similar titers. We also noted that in many notebook entries suggested additional reagents not already available to the reagent list, that it reasoned might improve specific cost. There were so many lab notebook entries, and GPT-5 had so many reagent suggestions that we asked GPT-5 to analyze and rank order its reagent suggestions. Sourced those reagents that were commercially available and added them to the list of reagents available to them. Dude, this is the freaking stuff, man. That is so awesome. In step 2, although GPT-5 was able to make use of the additional reagents, it chose to do so relatively sparingly. Nevertheless, GPT-5 achieved both lower reaction costs and higher titers versus step 0 and 1. In total, in step 2, 64 plates tested with data, a series of process improvements to our CFPS workflow to improve reaction predict. What does that even mean? In particular, we switched to a different SFGFP plasmid design and improved our cell-free lysate preparation process. Okay, we definitely have to go through the supplementary material. That's certain. Oh, this is a. Oh wow, this is cool. Wait, I don't really understand this plot. What are these clouds? And then what we're plotting here is titer versus reaction cost. That is so cool. So what you want is really high titer and really low reaction cost. So yeah, you want to be like right here. That's amazing. Oh, that's the Pareto frontier. Okay, so here they gave it tools. Okay. In step 3, we significantly expanded both the tools the model could access and the experimental metadata that we provided GPT-5. In particular, we provided access to the internet, computer, data analysis packages. We also provided the model with the preprint on by Olson et al., including supplementary. The model was able to access relevant scientific literature online. Finally, we exposed the model to additional potentially useful metadata, including raw fluorescence values, titers from positive and negative controls, reagent lot numbers. Oh, yeah, that's cool, dude. Reagent lot numbers. What if there's signal in that? That's funny. I love the lot numbers. Bro, when I sat there taking those pictures, oh man, I got comfortable with those lot numbers. Liquid handling errors, quality of fit metrics for per plate standard curves, instrument protocols and parameters, and actual CFPS incubation times. Oh, damn, bro. See, I was thinking about this. I was thinking about this like so much. Is like, yeah, and the system could be smart enough to know or learn like if there's a signal with variation in actual incubation time. That's funny. Together, these changes, along with improvements to the DNA template and cell lysate, both reaction cost and titer, with the model making more use of the additional reagents. Note that we opted to forego heuristic scoring in this step. I don't know what that means, honestly. Top performing reaction conditions from step 3 were K means clustered to select a diversity of reagent compositions, down selected by the... Is this the methods section? Is that what we're in right now? Oh, results. Okay, so they're describing a down selection pipeline. They tried tons of recipes, found the ones that looked best, then picked a diverse... Oh, okay. Basically, yeah, so they try to find... Okay, yeah. They try to find the ones that had good results, but were different. And then they retest in formats that behave differently. Tubes and 90s. So they picked from the different clusters, but the best in there. K-means is a way to group points that are similar. Each reaction condition... K-means partitions the top into K groups so that conditions within a group are compositionally similar. Because the top performers often aren't 50 totally different ideas. They might be minor tweaks. If you just pick the top 20 by score, you might accidentally pick the nearest duplicates. Yeah. Cluster them, select representatives, down select, keep the ones that are best, then retest. Why retest? That's what I want to know. Why retest in different containers? They're warning that titer depends on oxygenation and surface area to volume. Bro, surface area to volume. Yo, Bruce Heyer would be proud, man. Surface area to volume. Okay, that makes sense. I'm glad that I asked because now I understand exactly what's going on here. Okay. Step 4 was performed by the GPT-5. Dude, why do they have to say this? It's like, I guess they got to say it somehow. It took him 6 months to do 30,000 reactions. That is so cool, man. The GPT-5 driven autonomous lab improved CFPS specific cost by 40% versus... We benchmarked the performance of the reaction composition of the autonomous lab against the best performing composition RF opt from Olson et al., the current state of the art. Sought to reduce reagent costs, the cost of all other than cell lysate and DNA template. In light of their substantial reduction However, cell lysate now account for 90% of total CFPS reaction costs in state of the art. Interesting. Accordingly, we focused on optimizing total reaction cost. The sum of reagent. Indeed, we were able to further reduce reagent, lysate, and DNA costs. Total reaction costs of, what is this, CFE? What does CFE stand for? Cell-free something, but I don't know what. Okay, yeah. The largest retail unit. Yeah, so it's like the yeah, okay, that makes sense. So like if Sigma sells a gram, 10 grams, and 100 grams, they're choosing the cost at 100 grams. That's an interesting thing that they have to mention. I never thought about that. The autonomous lab was able to improve upon state-of-the-art by producing superfolder green GFP at a specific cost, blah, blah, blah. The composition obtained from the autonomous lab reduces it by 40%, increases... Okay. Although reagents only cost was not an explicit model objective, we also reduced reagent costs by... Wow. Wow, wait, why is the titer 10 times bigger? I mean, the titer is so much better in these bigger tubes. That's pretty crazy. Man, reaction geometry. What a cool phrase. Reaction geometry. Reported errors and... Okay, yeah. Reported errors for 8 replicates and 4 for 96 and 2. Okay. Look at these two have crazy deviations. But in general, it seems like the deviation... Well, no, but it's not exactly accurate, because what you need to do is look at the ratio, because like these are relatively less deviation overall with respect to the titer. And I imagine the trend holds there too. So for some reason, Deviations are just always higher in the 384 well plates. Here we go, the pedantic model. I don't know what the hell pedantic is, honestly. A key technical hurdle in the use of autonomous labs for scientific experimentation is ensuring that the AI models generate valid experimental designs that can be physically performed on the available equipment. We use Pydantic, a data validation library, to formalize the shared contract for experimental design, encode physical and logistical constraints, and define the schema from which catalyst protocols are generated and experimental results are returned and analyzed. We imposed key experimental design constraints, including plate format and layout standards controls, replication factor, and allowed reagent volumes. We also parameterized the available CFPS reagents, stock concentrations, chemical composition, and unit costs. GPT-5 interacted with the Pydantic schema to produce a JSON file representation of a CFPS experiment, a 384 well plate with specified reaction compositions. The Pydantic schema ensures that it produces an experimental design that is both syntactically well-formed for catalyst protocol generation and physically executable on the lab. The plate JSON file serves as a convenient abstraction and interface between the LLM, which is focused on experimental intent and design, and the cloud lab, which handles all the complexities of physical implementation. Okay, here we go. This is going to be good. Useful lab notebook entries. As discussed above... Wait, hold on. GPT-5's lab notebook entries summarized prior data, interpreted results, and proposed new hypotheses for testing. In particular, it suggested new reagents that it deemed potentially useful to reduce specific cost, accompanying rationale for their inclusion in target concentration ranges to try. For instance, it noted that nucleotide triphosphates were a dominant cost in CFPS reactions and prioritized reagents that it reasoned could support noted that NTPs were a dominant cost in CFPS reactions and prioritized reagents that it reasoned could support NTP regeneration or NTP synthesis. For real, the future is now. This is like crazy. Thus, GPT-5 anticipated certain reagents such as nucleotide monophosphates, potassium phosphate, and ribose that were critical to the reagent cost advances. Oh, described by Olson et al. Note that GPT-5 did not list nicotinamide in its prioritized reagents to add. However, it did try and ultimately used it in all top-performing reaction compositions. Of more than 20 additional reagents, several were components of one or more of our top-performing reaction compositions, including nucleotide monophosphate, glucose, potassium phosphate, dilithium acetyl phosphate, adenosine guanine catalase, and oxaloacetic acid. The model also suggested various enzymes that it reasoned could be helpful in NTP regeneration or synthesis. Wait, are these the recipes? These are the recipes, dude. So if you just throw this stuff together, millimolar unless otherwise noted. What is tyrosine? I mean, yeah, obviously the lysate. So, but okay, so I guess. Well, how did they do the purification? Okay, tool use. Heuristic scores from grading of experimental designs not. Wait, heuristic scores from grading of experimental designs did not obviously correlate with superior design. At each step, we ranked and selected all Pydantic validated generated experimental designs using one of three scoring methods. Heuristic, in which another LLM with a principal investigator persona. Yeah, well, it's good that they did that because, you know, critique the design and justification. Oh, I'd love to see that. That's just so funny. Random, in which only Pydantic. Well, C is probably interesting. Scoring step was used. Zero heuristic. Oh wait, what? Why didn't they do all of them on all of them? In 6 steps spanning 6 months, GPT-5 was able to find reaction compositions that surpass soda in specific cost and titer. The top performing reaction compositions differ from soda in both reaction composition and component concentration. When prompted to synthesize insights from steps 3 to 5, GPT-5, guided by human input and subsequently checked with our own median titer calculations, made several observations and potential rationales that we found plausible but require further experimental validation. Adding cheap HEPS buffer has an outsized impact on the specific cost by preventing yield collapse from pH changes. Generally, it found that higher HEPS concentrations increased titer and thus decreased cost. GPT-5 also noted that there is an interdependence between other reaction components and the optimal amount of buffer. For example, in step 4, reactions without added HEPS just the HEPS in the base buffer had a median titer nanoliters. Phosphate must be buffered with mono and dibasic potassium phosphates within an optimum concentration and pH range, lest titers fall precipitously. NTP is not cost effective. In step 5, reactions with adenosine at a median titer of spermidine, the low-cost reaction component for which addition asserted that polyamides such as spermidine stabilize nucleic acids and ribosomal function, thereby improving transcription and translation efficiency. Because it lowered titer, or increased titer, but it didn't. It low cost, so it had okay. Sodium hexametaphosphate did not have beneficial effect. Reaction costs are dominated by cell lysate and DNA template, but improvements in titer from buffering, pH control, or from addition of spermidine can materially improve the overall specific cost. To evaluate whether the CPU developed here could be used to produce other proteins, we tested it with a panel of 12 proteins, each of which wait, beta, not galactosidase, beta glucosidase. That probably cleaves glucose bonds. I think two glucoses is but this is the yeah, cellobios. Why would you want to make that? I love these. Isn't that fun? I like that model kit. That model kit looks good. Oh, I'll be right back. Our work demonstrates the real-world application to address an open scientific problem. We used hold on. Okay. All right, let's keep going. What's the holdup? Wait, actually. Okay, here we go. Our work demonstrates the real-world application of an autonomous lab to address an open scientific problem, the simultaneous cost and tighter optimization of CFPS. We used GPT-5 to plan and design experiments in the lab to execute those experiments. Human intervention was primarily limited to preparation, loading, and unloading of reagents and consumables. We were able to improve upon the state of the art to produce SFGP at a specific cost of, representing a 40% reduction in reaction cost per gram of protein. That's amazing. Although the CFPS reaction costs quoted here are not directly comparable to the prices of commercially available CFPS kits, it is notable that the NEB Express kit, as one example, has a list price of $800,000 per gram of protein. Yeah, what the hell? I don't think that can't be right. $800,000 per gram of protein produced. Importantly, our GPT-5 driven autonomous lab was able to reduce the reaction costs while increasing titer. We anticipate that additional optimization for particular reaction geometries, particular reaction volumes, or the protein of interest could further improve the production. Use of GPT-5 for experimental planning and design enabled data generation at a scale sufficient to support reinforcement learning. In total, we ran over 580 microtiter plates, suggesting 36,000 or tested 36,000 compositions, and generated almost 150,000 data points in 6 months. With steps 3 to 5 just taking 2 months. We believe the scale and speed of successful experimentation could not have been possible without LLM Autonomous Lab. I mean, that's just a fact, man. There's no way you could do this otherwise. In autonomous labs, experimental costs are dominated by reagents and consumables, not personnel. We believe that our use of an autonomous lab to optimize CFPS is a harbinger of things to come. Particularly apropos, one of the many potential applications of this work is a cloud laboratory running low-cost CFPS workflows as a useful complement to the many AI-based protein design tools available today. We chose not to use GPT-5 to write the Catalyst protocols responsible for executing the rack workflows. Indeed, all Catalyst protocols were written by... Okay, see, that's what I wanted to know. That's what they hide in the discussion. Instead, we constrained GPT-5 by the pedantic schema to produce experimental designs within a predefined operational envelope of Ginkgo's cloud laboratory. We believe that this constraint appropriately focused the GPT-5 autonomous lab on exploring relevant regions of the biological parameter space rather than on writing, attempting to run, and debugging rack-compatible protocols. We intend our work to open the aperture of what Thomas Kuhn calls normal science. Bro, I gotta reread that book. I found a picture of myself. No, I found a picture, not of myself, that I took in like 2021 or 2020. It was an omelet. It was here back in the freaking patio, and I was reading that book. I gotta reread that book. Where is that book? I love the cover. What is it called? The Structure of Scientific Revolutions. That's not the cover that I'm thinking of. No, no, no. No, no, no. Oh, wait. That's the one. That's the one that I have. I love that cover. All right. A common concern with the use of the current generation of LLMs for scientific research is the possibility of hallucinations, in part due to the pedantic. We found that hallucinations were quite rare. Less than 1% of executed designs were flawed. In one plate design, the model attempted to overwrite the required volume. So my question is, you know, how specific, how how specific is specific cost and titer likely to be with respect to the protein being produced in cell-free protein synthesis? How specific is reaction composition to the protein of the produced protein? How protein specific is titer? mRNA. Protein properties: size, folding rate, disulfide. Remember? It's curious that they didn't focus on lysate. Why didn't they optimize the lysate? In the immediate future, we believe work such as that described here will become common across a wide range of biological, biomedical, and even chemical science. In just a few years out, as reasoning models grow ever more powerful and modular laboratory automations become pervasive, autonomous lab experimentation will increasingly supplant traditional lab work, allowing scientists to focus on harder and more fulfilling work. Oh, here we go. Methods and materials. Okay, the pydantic schema comprised 3 primary models to define the interface between the experimental design and the cloud lab. The plate model represented the complete experimental design, including plate geometry, layout, sample definitions. The plate model represented the complete experimental design, including plate geometry, plate layout, sample definitions, and high-level composition metadata such as reserved columns and the number and placement of controls. The sample model represented an individual cell-free protein synthesis reaction, capturing the experimental condition at the well level, validating the desired replication for each sample. The reagent list model specified the set of reagents and the associated volumes for each level. At this level, the availability of reagents and permissible reagent volumes were validated against inventory and constraint rules. For each iterative step, we generated 2x the target number of designs from GPT because not all experiment definitions pass pydantic validation. Wait, they open sourced it? Dude. Okay, we gotta read all of this. But I want to go to 7.8 because I gotta see that. They're using UV, bro. Who doesn't use UV, man? UV is the freaking. Where are the units, bro? See, this is the crazy thing about Python, man. Is Emerald Cloud Lab still around? The crazy thing about Emerald Cloud Lab though is the Mathematica drive. It's all Mathematica. You could tell that based just the way it looks, it's all Mathematica. Click to copy, bro. That's crazy. This is the prompt. You will predict optimal reagent volumes for a cell-free protein synthesis experiment. The goal is to maximize synthesis of the erythropoietin protein. That's curious. The current list of reagents is available. Their initial stock concentrations, their components, and their cost per milliliter is as follows. Our desired reaction volume is 20 microliters. Each well is pre-filled with 6 microliters of cell lysate. Why is the cell lysate so expensive? Why do they have it under a promoter? Your task is to provide values in nanoliters for each that would optimize synthesis of erythropoietin. I don't believe the code in here that actually does anything with Catalyst is open source. I think that... Well, that's cool. Version control of genetic sequences. All right, so wait, what else do we have left here? Oh, here we go. Cell lysate preparation. I actually do need... Lysate was generated from a fermented E. coli strain. We grew cells overnight at 30 C and LB to prepare starter culture. The following day, we combined all... The following day, we combined all starter cultures and confirmed OD. We aseptically charged inoculum to Ginkgo's FM39 production medium, supplemented with isopropyl beta... What? To induce... Oh. Wait. T7 RNA polymerase expression? Oh, I think that it's not like that... Whatever T7 bacteriophage thing that we saw with the promoter. I think that that's just the specific sequence for a specific type of RNA polymerase. I don't think it's like regulated or anything. But I think the next step here, I mean, there's so many places they could go. First off, the lysate, other proteins, non-protein chemical synthesis, like reaction, like doing mechanisms um, to synthesize some molecules. But then also scale up, obviously. How literally, if we start to mess with like putting it in a bioreactor, a cell-free bioreactor, they got the recipe down. Does it scale? That's an obvious question. Um, um, you know, can you do something to that does require genetic controls? Feedback, whatever. Into 5 liter bio. Okay, so they did. This is how they're making their lysate. For fed batch style fermentation with temperature, oxygen, and anti-foam control. Hate these companies. Inquire for us to tell you that it's $1 million. Why not just tell me it's $1 million? Once fermentation was complete, we harvested and pelleted cultures. We discarded the supernatant and washed the pellet 3 times in the harvest lysis buffer. After the third wash, the pelleted material was flash frozen in liquid nitrogen and stored at -80. We removed the washed fermentation pellet from the freezer and thawed at room temperature. We supplemented the harvest lysis buffer with DTT to the thawing pellet and agitated it to ensure a homogeneous mixture. Once fully thawed and homogeneous, we lysed the material with a Dude, I hate fucking Firefox, bro. Firefox's downloaded settings are so stupid. Okay, wait, hold on. In the harvest lysis buffer, we lysed it with this homogenizer. So what is that? We need to know what that is. All right, let's just download it. I don't care. We clarified the resulting lysate twice by we allocate. Oh, so they don't lyse it with they don't do chemical lysis. That's crazy. Isn't she like the isn't she one of the founders? Yeah, she co-founded it. That's what I thought. Bro, she rules. This is the wrong person. This is the person. Okay, cool. Okay, so I think that that's it for right now. Uh, that was really interesting to read.